Welcome back to part 2 of our protein language model introduction videos. In part 1, we introduced protein language models and saw that proteins are sequences of amino acids, and we can treat these sequences like sentences in natural language. We saw that a protein language model is usually a transformer encoder that turns each amino acid into a context-aware embedding, and that by training on large datasets of sequences and predicting masked amino acids, the model learns rich representations that capture structural and functional information. And we discussed how we can compress residual embeddings into a single protein embedding that we can use for downstream prediction tasks. In this second part, we will look more closely at the transformer network encoder architecture and how the amino acid embeddings are actually processed. Here you see the architecture of a transformer network encoder. There are 5 main building blocks that we will look at. First, how we compute the input token embeddings when we present a protein sequence to our model. Second, the attention mechanism that is used to update the token embeddings. Third, the feed-forward network. Fourth, the residual connections. And finally, we will look at layer normalization. We remember that we need to divide the input sequence into smaller subsections, the so-called tokens. For proteins, each amino acid is a separate token. Each unique token, or in this case amino acid, gets assigned to a unique token ID. The set of all possible tokens in the model is called the vocabulary. We need 20 tokens for the 20 standard amino acids, plus a few tokens for special tokens like the masking or classification token. We want to represent each unique token through a different vector that encodes what type of token we have. The dimension of the input vectors is equal to the hidden dimension d of the network, which is typically in the range of a few hundred up to a few thousand. Intuitively, we can imagine that the token embeddings for all possible tokens are stored in an embedding matrix of the hidden dimension d times the vocabulary size, which could be, for example, 23. When inputting a protein sequence into a transformer network, we map each amino acid to its corresponding token ID. We can then simply access the columns corresponding to the token indices. In protein language models, the entries of this embedding matrix are learnable parameters, which means the model can adjust the initial token embeddings during training. Additionally to what type of amino acids we have in the sequence, the model must also know where each amino acid is in the sequence. For this, we also need embeddings that encode the position of the tokens. This works very similar to the token embeddings. We assign to each token a position index, and then we get a position vector of dimension d for each position. We store one position vector for each possible position in a matrix. These position vectors can be learned in the same way as we've just seen for the token embeddings. Alternatively, we can use fixed embeddings by using predefined functions for computing the position vectors. Most commonly, sinusoidal positional encodings are used. A third and slightly less commonly used way of encoding positions is using so-called rope embeddings, which stands for rotary positional embeddings, where we don't compute position vectors, but we rotate the token embeddings depending on their position. You can find a detailed explanation of rope embeddings in our ESM2 video. Here we will focus on learned or fixed sinusoidal positional embeddings. We now want to combine for each token the position and the token type embeddings into a single vector. We simply add the position vectors to the token embeddings, resulting in token representations that encode both the type and its position within the sequence. For example, the resulting vector at the 4th position stores the information that we have the amino acid methionine at the 4th position. It may seem a bit counterintuitive that instead of concatenating the 2 position and token embedding vectors, we compute their sum, since this could result in a loss of information. However, the transformer network can easily learn to separate the positional information from the actual token embedding during the training process, and thus this allows it to store both information in an efficient way without information loss. Now we know how we get initial input embeddings for all amino acids in a protein sequence. For each token, we have a vector containing information about what type of token we have and where it is located. Next, we will look at how the attention mechanism is used to update these embeddings by incorporating sequence context from the other embeddings. The core of the transformer encoder is the self-attention mechanism. Intuitively, self-attention does the following for each amino acid. It compares the embedding of the amino acid that we want to update to the embeddings of all other positions in the sequence. It computes importance scores indicating how relevant each other position is, indicated here by the different darknesses of the lines. And then we normalize all these scores so that we have scores between 0 and 1 that add up to 1. And then we take a weighted average of all residue embeddings using these importance scores. The result is a new vector for that position that includes information from everywhere in the sequence. This update is computed in parallel for all positions in the sequence, which makes training efficient. For proteins, these updates are useful because a residue can attend not only to its neighbors, but also to residues that will be close in the 3D. But how do we actually compute the importance scores that we need to update the token representations? This is done using the attention function. To update embedding vector EI at position I, we use information about the vector at position I itself and about all the other vectors in the input sequence E1 to En that are stored in the matrices K and V. We will now take a closer look at this function and we will see step by step what is happening. You see again the definition for updating embedding vector EI using the attention function. The input of this function is the vector EI itself and the matrices K and V, which contain in their columns all the embedding vectors E1 to En, which means they contain information about the whole input sequence. You can see that the first part of the attention function consists of the softmax function, where we have inside the softmax function the product of vector EI transposed with the matrix K. This means that we are calculating the scalar product of EI with all the other n vectors, which results in a vector of length n that contains our importance scores, which are then scaled between 0 and 1 by applying the softmax function. Multiplying this vector with a v transposed is equal to calculating a weighted sum of all vectors E1 to En. But let's go through it step by step. Let us assume we want to update the second embedding vector E2. First, we calculate inside the softmax function the multiplication of vector E2 transposed, the representation of the second amino acid of the sequence with matrix K. We see that K contains all vectors E1 to En in its columns. Hence, we calculate the scalar product of E2 with all vector representations of the input sequence. We can interpret this as comparing vector E2 with all the other vectors. This results in a single value for every scalar product. And for vectors that are more similar to each other, this results in higher numbers. Next, we scale all values of this vector by dividing through the square root of d, which is the dimension of the representation vectors. The reason for this is that the higher the dimension d is, the larger the magnitude of the scalar products can become, and by dividing by the square root of d, we account for that. Then we apply the softmax function. It converts all numbers of the vector to values between 0 and 1 that sum up to 1. So this results in a vector with n entries between 0 and 1. And you can see that values that have been higher are still higher, and values that were low or negative are transformed into values close to 0. These are now our importance scores that we want to use to update vector E2. Finally, we multiply this row vector by the matrix V transposed, which has in its rows the token representations E1 to En. We can see that this means that we calculate the first entry of our vector, which is equal to 0.1 with vector E1, the second entry 0.55 with vector E2, and so on. We are calculating a weighted means of all the vectors E1 to En, and the resulting vector is our new updated representation of the second amino acid. I did not tell you the whole truth about the attention function yet. Instead of applying this attention function directly to the vector representations E1 to En, we first compute linear transformations for all input vectors by multiplying them by matrix W. You can see that the input vectors EI that we want to update are transformed by multiplying them by matrix WE. The vectors stored in the matrix K, which we compare with the vectors EI, are also multiplied, but by a different matrix WK. The resulting matrix is named K tilde. and the vectors in the matrix V, which we use to compute a weighted sum of all representations, are also transformed by multiplying them by another matrix WV. The reason for doing these linear transformations of the token representations is to project the original embeddings into different subspaces that are more suitable for capturing different types of relationships between the amino acids. This allows the model to understand the different meanings and rules of how amino acids are arranged in proteins. The entries of these matrices W are adjusted during the training of a transformer network. The attention function is the most crucial part of transformer networks, how the model uses the attention mechanism to update its embedding. This is what makes large language models like ChatGPT or protein language models like ESM2 so powerful, processing the whole sequence at once using the attention function. In practice, we use multi-head attention, which means that we apply the attention function multiple times but with different parameters. Each of these attention functions are called a head, and for example, we could have 8 of such heads in one attention block. So let's have a look at how these heads look like. To update the embedding vector EI with head number j, we apply the attention function to this vector. Here it is important to note that in every head we use different matrices WE, WK and WV to transform the embedding vectors that we use as the input for the attention function. If we wouldn't do this, we would simply apply the same attention function h times. After we passed vector EI through all h attention heads, we want to combine the resulting vectors in a single vector that is then our updated representation for the vector EI. We can achieve this by simply concatenating the resulting vectors, which simply means that we can attach the vectors that are the results of calculating all h attention functions. And finally, we multiply the resulting vector by matrix WO, which is simply a linear transformation. When we do this, we have to keep in mind that in the end we want to have a vector that has dimension d again, which is the same dimension of the vector EI that we used as the input for the multi-head attention function. So let's look at the dimensions of the matrices and the resulting vectors. In order to get a vector in the end that has the same length d as the initial vector EI, we want that each attention head produces a vector of reduced size. This reduced size is d divided by the number of attention head h, and we call this reduced dimension m. To achieve this, the matrices WE, WK and WV must have dimension m times d, because multiplying a m times d matrix by a d-dimensional vector results in a m-dimensional vector. If we then concatenate those h vectors of dimension m in the step below, we get a vector of dimension d again. And if we multiply this vector by a d times d matrix WO, we finally get a vector of dimension d, which is what we wanted. Multi-head attention allows the model to focus on different parts and aspects of the input sequence for each attention head. Each head can specialize in attending to different types of information or patterns within the input, leading to more informative representations. And due to the reduced dimension of each head, the total computational cost of the multi-head attention is similar to the computation cost of single-head attention with full dimensionality. Let's do a short repetition of what we've looked at so far. First, we generated for each amino acid in a protein sequence an embedding that encodes the type of amino acid and its position within the sequence. Then we used the attention function to update each embedding, incorporating information from the other amino acid embeddings in the sequence. We are now looking at the feed-forward block, where we update each embedding separately using a standard multi-layer perceptron, a feed-forward neural network with one hidden layer. This means that this time we do not use information about the other amino acids in the input sequence, but only about the token representation itself. This feed-forward neural network is defined by this function. We calculate first the multiplication of a matrix W1 with the vector that we want to update, and then we add the vector B1. The entries of the matrix W and the vector B are learnable and adjusted during the training of the transformer network. We apply an activation function like the ReLU function to the resulting vector, which sets all negative values to 0 and leaves all positive entries unchanged. Then we apply another matrix multiplication to the resulting vector with the matrix W2, and we add another vector B2. we apply the same function to all embeddings separately. Typically, the matrix W1 has the dimension 4D times D, which means that we map our d-dimensional vector to a vector of 4 times this dimension. Matrix W2 has dimension D times 4D, which means we map the embeddings back to vectors of dimension D. The role of this feed-forward block is that it takes each token's mixed attention information and transforms it into a hopefully richer, more useful representation that can be better processed by subsequent layers. When we look at our transformer encoder overview figure, we see that we have almost discussed all building blocks. Only the residual connections and layer normalization is missing. Adding a residual connection simply means that after calculating an updated embedding vector at position I with the attention function, we simply add the original vector EI to this updated vector. This has the advantage of making it easier to train the encoder by addressing the vanishing gradient problem. And in the same way, we have a residual connection around the feed-forward block. Residual connections ensure that the gradients do not become zero or very close to zero during backpropagation when training the model for very deep architectures. On the other hand, it ensures that we keep the information from the input vectors and the model can still use this information very easily. Let's have a look at layer normalization that is used to normalize the updated embedding vectors. We will look at how layer normalization works by looking at a small toy example. For practicality, let's assume the dimension of the vectors that we get as the output of the multi-head attention is very small, in this case 3, and we assume that we are processing a sequence that consists of only 2 tokens, and we are currently only processing one sequence. Then, the output of the attention function is 2 token representations E1 and E2, both of dimension 3. We can normalize each of those 2 vectors by calculating the mean of both vectors and the standard deviation of both vectors separately. So we see the first vector has a mean of 2, the second vector has a mean of 1, and both vectors have a standard deviation of approximately 2. Now we normalize each vector by subtracting the mean μ1 from E1 and dividing by the standard deviation sigma1. This gives us a vector that has mean 0 and a variance of 1. And we do the same for the second vector using μ2 and sigma2, resulting in normalized vectors. Layer normalization now allows the model to rescale those normalized vectors by adding learnable parameters gamma and beta that have the same dimension as our vector representations. We calculate the Hadamard product of gamma with the normalized vectors. This means we multiply the first entry of gamma with the first entry of the normalized vector and use this as the first entry of the resulting vector, and so on. Then we also add the vector beta to the resulting vector. Here it is important to note that we rescale all embedding vectors of the sequence using the same parameters gamma and beta. This rescaling with gamma and beta gives the model the flexibility to learn and adjust the scale and the shift of the normalized data, allowing it to scale it to a distribution that is best for efficiently training the model. In our overview figure, we see that we apply layer normalization after the attention block and after the feed-forward block. We have now looked at the main building blocks of the transformer network encoder that is used in most protein language models. Instead of applying the attention block and the feed-forward function only once, we have multiple layers of these attention and feed-forward functions. We have seen that the first set of attention block and feed-forward block produces a numerical vector for each input token. These vectors have the same number and dimension of the vectors that we receive from the input embeddings and the positional encoding. So we can simply use these vectors that come out of the first layer of the encoder as the input for a second layer of the encoder that again consists of the same building blocks that you can see here, but with different learnable parameters. We do this to further update and improve the token embedding representations, and we do this for a predefined number of times. And the vectors that come out of the final encoder layer are our final token representations. In the first video about protein language models, we saw that we pre-trained protein language models using mask language modeling. Let's have a look at which parameters are actually trainable. So what can we adjust during model training to achieve better representations? If we use learnable token and position embeddings, we can adjust these values. Then we have the attention block. We have seen that in each head of the multi-head attention function, we have matrices WE, WK, and WV. All entries of these matrices can be adjusted during training, and also the matrix WO that we use to transform the concatenated vector has adjustable parameters. Then we have layer normalization, where we have the learnable parameters gamma and beta. Those parameters allow us to rescale the normalized embeddings. And finally, for the fully connected neural network, we have again matrices W1 and W2 with trainable entries, and also bias vectors B1 and B2 with adjustable entries. Then we have again learnable gamma and beta parameters in the layer normalization following the feed-forward block. And of course, we have different sets of all of these red parameters that you see here in each layer of the transformer network encoder. Before we train the encoder, we have to set the learnable parameters to some initial starting values. Typically, the bias vectors B are set to 0, the entries of the matrices W are typically set to small Gaussian distributed values that are distributed around 0. And for the parameters of the layer normalization, you would typically set all entries of gamma to 1 and all entries of beta to 0. This means applying beta and gamma to your function is initially not changing anything. You should now have a good understanding of how protein language models work and how they are trained. If you want to have a more detailed look at how the most commonly used protein language models work, we have put the links to these videos in the video description.